Hello everyone. Today we'll be solving EDEXCEL IGCSE Biology Paper 1B Jan 2022. Today we'll be solving from question number 6 to question number 10. The diagram shows part of the gut of a rabbit. The rabbit is a primary consumer, eats mainly grass and other vegetable materials. We can see A, the esophagus, B, which is the stomach. We can see C, which is the small intestine. We can see D, which is the large intestine. Name the parts labeled A, B, C, D. Part B. The gut of a rabbit has a large cecum and appendix. These contains bacteria that are able to produce the enzyme cellulase. Explain how these bacteria helps the rabbit with their diet of plant material. So because the rabbit has a diet of plant material, plant contains cellulase. Cellulose as in their fibrous portion. So the cellulase enzyme helps digest the cellulose into glucose, which is then used in respiration. Part C. The human gut has a cecum and appendix, but they are much smaller than those in the rabbit. Suggest why the human gut only has a small cecum and appendix. First of all, in humans' diet, there is not much of cellulose. We do not need to digest cellulose. So, because of that, we do not need the cecum. All right. And neither do we need the appendix in such a large proportion. C2. In humans, the appendix also acts as a store of useful bacteria. Scientists have discovered that patients who have their appendix removed are more likely to develop infections in the colon. Explain how having no appendix may increase the likelihood of bacterial infection of the colon. So basically, what happens is that once the appendix is removed, it also removes the useful bacteria that are produced and secreted by the appendix. All right. So this reduces the competition with the pathogenic bacteria, and pathogenic bacteria can survive better. They can multiply much more faster. All right. And as a result, they can basically cause bacterial infection. Question number 7. The diagram shows a food web from a woodland ecosystem. Now we can see the oak tree, which is the producer, and then we can see earthworm and woodlouse. These are primary consumers. So we'll just label them 1C. They are primary consumers. When woodlouse is eaten by centipede, second centipede becomes the secondary consumer. And when when the centipede is eaten by blackbird, the blackbird becomes tertiary consumer. When blackbird is eaten by sparrowhawk, it becomes the quaternary consumer. When the earthworm is eaten by the shrew becomes the secondary consumer. And when the shrew is eaten by sparrowhawk, it becomes tertiary consumer. However, if the woodlouse is eaten by ground beetle, then the ground beetle in this pathway, it becomes the secondary consumer. And that makes the shrew a tertiary consumer, which in turn makes the sparrowhawk a quaternary consumer from this perspective. All right. If the ground beetle is eaten by the blackbird, then it also becomes tertiary consumer. And when the blackbird is eaten by the sparrowhawk, it becomes tertiary consumer, so, quaternary consumer. So, all right. Now, moving on to the question, which organism in the food web is the producer? So we can definitely say the oak tree is the producer. Which organism acts as both secondary consumer and a tertiary consumer in the food web? So we can see the shrew is acting as both a secondary and a tertiary consumer in the food web. So both secondary and tertiary. All right. However, there is a problem. The problem is shrew is not included in this particular list. We see blackbird, earthworm, and ground beetle, sparrowhawk. So if we consider that the sparrowhawk, the blackbird is eating the earthworm. In that particular consideration, blackbird becomes secondary consumer, and the blackbird eating the ground beetle becomes tertiary consumer. So blackbird will be the correct answer. A. Part B. The amount of energy transfer changes as you move along a food chain. The data comes from an ecosystem containing producers, primary consumers, and secondary consumers. The light energy reaching the producers is 7.1 into 10 to the power of 6 kilojoule per meter square per year. Explain why the plants cannot absorb all of this energy. So we know that a plant, reflects certain wavelengths of light, and not all of the light energy falls on the leaves. So not all of the light energy can be absorbed by the plant and by the chloroplast in the plant or the chlorophyll. Part 2. The table shows that energy is transferred between producer and primary consumer and between primary consumer and secondary consumer. A student states that energy transfer between producer and primary consumer is the most efficient. Determine whether the student statement is correct. So we can see the producer 8.7 into 10 to the power of 5 kilojoule per meter square per year. And then from the producer to primary consumer, it is 1.4 into 10 to the power of 4, all right, energy that is transferred. From there, all right, from there, secondary consumer gets 1.6 into 10 to the power of 3 kilojoule per meter square. Now, the student commented that a student states that energy transfer between producer and primary consumer is the most efficient. So let's see in terms of percentage. Part 2. The table shows that the energy transferred between producer and primary consumer and between primary consumer and secondary consumer. The student states that the energy transfer between producer and primary consumer is the most efficient. Determine whether the student statement is correct. Now we can see from producer to primary consumer, 8.7 into 10 to the power of 5, all right, and primary consumer 1.4 into 10 to the power of 4. So if we do the math, we'll have to do it in this way: 1.4 into 10 to the power of 4 divided by 8.7 into 10 to the power of 5 into 100, and that gives us a 1.61 percentage. All right, now for the primary consumer and secondary consumer, if we look at it, then we can see 1.6 into 10 to the power of 3 divided by 1.4 into 10 to the power of 4 into 100%. That gives us about 11.43%. Now, the student concluded that the energy transfer between producer and primary consumer is the most efficient. As we can see, the efficiency at that level is 1.61%. So it is much lower than that of the primary consumer to secondary consumer, which is 11.43%. Part C. Woodlice feed on dead and decaying plant material in the soil. The photographs show how a woodlouse can curl up into a ball. This behavior is an example of reflex response. State what is meant by the term reflex response. So basically, a reflex response is autonomic, it's automatic, it's involuntary. All right, it does not involve the use of a brain without thinking. Part 2. Give a reason why this reflex response benefit the woodlouse. Well, it protects the woodlouse from the predators, all right, and only exposes the hard shell of the woodlouse towards the predator, which make it unpalatable, all right, for the predator. Part 3. Describe how this reflex response could be evolved by natural selection. So, natural selection can give rise to this kind of reflex response by means of gene mutation. So whenever there is a gene mutation, it leads to variation, and probably there was this particular variation where the wood lice that roll up, they were not eaten, they survived, so they were able to reproduce, they grew until their adulthood, and then they were able to reproduce, and when they reproduce, they pass on their alleles for the rolling behavior to the next generation. And when they passed on the alleles for the rolling behavior to the next generation, and only the wood lice that were able to roll up were able to survive, then that became that became the norm within the natural population, and they have evolved. Question number 8. The rate of photosynthesis is affected by different factors. One factor is the concentration of carbon dioxide in the air. The percentage of oxygen in the air is 21%. This is equivalent to a concentration of 210,000 parts per million. The percentage of carbon dioxide in the air is 0.04%. Calculate the concentration in parts per million. So basically, 0.04%, it means that there is 0.04 in 100. So in 1 million, it will be 1 million divided by 100 multiplied by 0.04, which gives us an answer of 400. So 400 ppm. Guys, if you haven't understood how the response were brought about, because we had 0.04%, we had 0.04% of carbon dioxide, and that actually, whenever we put the percentage, it means out of 100. So we had 0.04 out of 100. Now the 100 is represented by a million when writing parts per million. So when there is 1 million and we divide it by 100, and then the answer, resulting answer, we multiplied by 0.04, we get uh, the parts per million answer. So 400 ppm is the answer. Part B. The graph shows the effect of increasing concentration of carbon dioxide in the air on the relative rate of photosynthesis at different temperature. Concentration of carbon dioxide increases, relative rate of photosynthesis increases on the y-axis. Part 1. Describe the effect of increasing the concentration of carbon dioxide on the relative rate of photosynthesis at 5 degrees Celsius. We can see when at 5 degrees Celsius, even at 5 degrees Celsius, increasing carbon dioxide concentration increases rate of photosynthesis, or a relative rate of photosynthesis. So we're going to comment increasing carbon dioxide increases relative rate of photosynthesis. However, it soon levels up. All right, it starts to level up as it reaches 1.1 relative rate of photosynthesis. Describe how the effect of increasing the concentration of carbon dioxide on the relative rate of photosynthesis changes when the temperature is increased. So from this particular graph, we can see that as we keep on increasing the temperature, the increase in temperature increases the rate of photosynthesis, relative rate of photosynthesis. uh, as we keep on increasing the temperature, we can see most efficient rate of photosynthesis occurs at 35 degrees Celsius, and the slowest rate of photosynthesis occur at 5 degrees Celsius. Now, along with that, we can also see that increasing carbon dioxide concentration also increases the rate of photosynthesis throughout the experiment. So, guys, the question was, describe how the effect of increasing concentration of carbon dioxide on the relative rate of photosynthesis changes when the temperature is increased. If we notice that at 0.03% concentration of carbon dioxide, no matter at which temperature, they all start at the same relative rate of photosynthesis. So at 0.03% concentration of carbon dioxide, temperature is not the limiting factor. There is something else other than the temperature which is the limiting factor. All right. However, there is a steepest increase in photosynthesis that occurs at 35 degrees Celsius. As you can see, at 35 degrees Celsius, the rate of photosynthesis is the highest. And increasing CO2 increases relative rate of photosynthesis at all temperature. As you can see, increasing the CO2 concentration, increase rate of photosynthesis at 5, even at 15, 25, and also at 35. So moving on, at CO2 of 0.03%, all right, 0.03% CO2, temperature is not the limiting factor at that particular point. That's why increasing temperature has no effect. Explain the effect of increasing temperature from 5C to 35C on the relative rate of photosynthesis. Okay, so explain the effect of increasing the temperature from 5 degrees Celsius to 35 degrees Celsius on the relative rate of photosynthesis. We can see that increasing temperature increases the relative rate of photosynthesis. There is no question into it. Why is it? Because there is more, as as there is an increase in kinetic energy. As the temperature increases, there is an increase in kinetic energy supplied to molecules. So there is more frequent collision between the substrate and enzyme molecules. The temperature becomes a limiting factor at 5 degrees Celsius, because you can see at even the increasing carbon dioxide concentration, it does not increase the rate of the reaction significantly, the rate of the photosynthesis significantly. Part C. The scientists who carried out this study concluded that the effect of increasing the concentration of carbon dioxide on the rate of growth of plant is dependent on temperature and also on the minerals that the plants can absorb. Explain how lacking unnamed minerals might affect the plant growth. So basically, we know that nitrate is needed for amino acids and protein. And without protein, the plant will have stunted growth. The plant will have yellow leaves, because chlorophyll is itself a protein. And then for magnesium, all right, magnesium is needed for making chlorophyll. So without magnesium, we're going to see chlorosis or yellow leaves. Explain how a named factor can affect the rate of photosynthesis other than carbon dioxide concentration, temperature, and mineral absorbed. So we know that photosynthesis is carried out by light energy. So increasing light intensity also provides more energy that is needed for photosynthesis. All right. And thereby, the rate of photosynthesis can be increased. Question number 9. Alkaptonuria is an inherited condition caused by the presence of recessive alleles. State what is meant by recessive alleles. So basically, recessive alleles are only expressed when they are homozygous, when both copies of the alleles are present. Part B. Alkaptonuria is first diagnosed in children when it is noticed that they produce very dark urine that turns black when exposed to air. A woman and a man do not have alkaptonuria. They have a child who has the condition. The woman and the man are expecting second child. Draw a genetic diagram to show the genotypes of the woman and the man, the gametes they produce, and the possible phenotypes and genotypes of the second child. So first of all, parents in this case, we can understand that they're both carriers. So it's going to be capital A, small a, capital A, small a. And then the gametes that they produce, they're going to be either any one of those, capital A, small a. And then offspring. The offspring genotype will be a combination. Part 2. Calculate the probability that the second child is male and does not have the condition. Now we know that having a child, a male child with no condition, so male and does not have the condition, so no alkaptonuria. So first of all, we'll have to understand that 50% of the child that are born are, they're gonna be male. So 1/2, and then out of that, all right, three will have no symptoms. So 3/4 will have into 3/4. So 0.5 into 0.75. So the probability is 0.375. Part C. Alkaptonuria is caused by the body being unable to break down the amino acid tyrosine and phenylalanine. This leads up to a buildup of a toxin that causes damage to joints and tendons and can lead to the heart valve damage in later life. A new drug treatment is being tested that can slow the damage to the joints and tendons. Scientists selected 40 adults who all had alkaptonuria. They placed each patient into one of two groups. One group was given the drug treatment, and the other group was acted as a no, control. The scientists then compared the symptoms of the patient in each group after 3 years. They describe what is meant by control group. So basically, a control group is a group that does not they get placebo medicine. They do not get the treatment. Part 2. The table compares the control group with the drug treatment group. It shows that numbers starting and completing the trial are those showing harmful effects. It also compares the improvements in two symptoms of alkaptonuria. Number of patients starting the trial, control group 20, drug group 20. Number of patients completing trial, 17 and then 16. Number of patients showing adverse effect, 0. And drug group 2. Number of patients that died, 0. Drug group 1. Decrease in time taken to stand up and walk 3 meter in seconds, 0.54, 1.33. Increase in the distance in meters walked in 6 minutes, 6.7. Drug group 51.5. Evaluate whether the new drug should be recommended as an effective treatment for alkaptonuria. When we are asked to, all right, when we are asked to, whenever we are asked to evaluate a particular drug information or something like that, we must understand that we must see the presence of a control group and a drug group. So since there is a control group, so we can say that this is a valid study. All right, then we can see in the drug group there is a improvement in the increase in distance in meters walked in 6 minutes. We can also see a number of patients showing adverse effect. So the question was, evaluate whether the new drug should be recommended as an effective treatment for alkaptonuria. We can see in the control group we had 20 and in the drug group we had 20. The number of patients that completed the trial we had 17 and 16. However, there was a significant improvement. There was a significant improvement in the distance that in meters, all right? Distance in meters that walked in 6 minutes. For control group, it was only 6.7, and whereas the drug group, it was 51.5. So, yes, the new drug can be recommended, all right, because it improved distance, improved distance that is walk. However, we can also say that the new drug should not be recommended because small group size, because 20 people does not necessarily mean an entire population. There is also other information, those that are missing from here. All right. The reason why we cannot recommend it to other people, not all of the patients finished study. We can see most of the patients did not finish the study. We can see there is adverse side effects. We can see there is adverse side effects. One patient died in the drug group, whereas we can see there is no patient that died in the not the placebo group. There is no information on sex and other health condition of the patients, so we cannot conclusively suggest that this is a good drug and can be used on another person. Part D. Other scientists have suggested that eating fewer proteins that contain tyrosine and phenylalanine would reduce the symptoms of alkaptonuria. So this is why eating fewer of these proteins may be difficult. Patients may not know which food contain these proteins, and the proteins can be present in most of the foods, so the patient cannot be like eating nothing. And proteins is required for growth and repair of other body tissues. Question number 10. Farmers sometimes use biological control to reduce the damage to their crops caused by pests such as insects. Which of these is an advantage of using biological control over chemical control? Biological control over chemical control. So, biological control, it lasts a short time. Biological control leads to bioaccumulation. Okay, bioaccumulation occurs in substances those that cannot be broken down by the environment. However, biological control are biological substances, so they can be easily broken down by the environment. Biological control can last for a longer period of time. That's because it's biologically produced. All right. Now the question says, which of these is an advantage of using biological control over chemical control? You see, biological control can be very, very specific, whereas chemical control are not specific. So it is specific. Part B, aphids are tiny insects that have very sharp mouth parts. They push these mouth parts into the phloem found in stems. They then feed on the phloem content. We can see the picture of an aphid. Name two substances the aphid obtain from the phloem. So, the phloem is rich in sugar. So we can say, the aphid gets sucrose, a lot of sucrose. And the phloem is also rich in amino acids. So the aphid gets amino acids needed for growth. Part 2. Explain how aphids feeding from the phloem of crop plants can lead to reduction in yield. So basically, whenever an aphid is feeding on a crop plant, the decrease the amount of sugar that they're absorbing, meaning the plant will be able to do less respiration and less energy will be produced within the plant. And less active transport can also take place because there is less amount of energy available. All right, the aphids are eating all the carbohydrate and also taking away the amino acids. So if amino acids are taken, that means less amount of proteins will be produced. and as a result, uh, you know it will also spread diseases as it bites a hole into that particular plant. So that can be also a reason why there will be a reduction in yield. Part C. Silver flies and hoverflies are two species of insect whose larvae feed on aphids. Scientists investigate the feeding behavior of these species in the laboratory experiment. This is the scientist's method. Place a single silver fly in a container. Place a single hoverfly in a separate container. Keep the container at 12 degrees Celsius. Put 30 aphids on each container. Count the number of aphids consumed each day for several days. Determine the mean number of aphids consumed per day. The scientists repeat the method at the two higher temperatures. The graph shows the scientists' result. Hoverfly and silverfly at 12 degrees Celsius. All right, we can see mean number of aphids consumed per day by the hoverfly is about close to 3, 2.8, and by the silverfly it's about 0.9. And we can see it increases as the temperature increases for both cases. Now the question: the scientists conclude that the hoverfly is the most effective biological control agent for aphids. Discuss the scientists' conclusion referring to the information in the graph and the scientist method in your answer. So we can see the hoverfly actually eats more insect, all right, per day compared to that of the silverfly. The scientists conclude that the hoverfly is the most effective biological control agent for aphids. Discuss the scientists' conclusion referring to the information in the graph and scientist method in your answer. Hoverflies consume more aphids at all temperatures, as we can see from the previous data. It always consumed higher number of aphids at all temperature. Therefore, fewer flies are needed to be used as a biological control. Both flies consume more at higher temperature. We can see both flies at higher temperature it is consuming more number of aphids. And the greatest difference, all right, is at 12 degrees Celsius, where we can see hoverfly consumes almost 3 times more than that of silverfly. As you can see, at 12 degrees Celsius, the hoverfly consumes about 2.8, whereas a silverfly consumes about 0.9. So you can see that is almost 3 times more than that. of the silver fly. Guys, thank you for joining the video and hope to see you in the next video. If you like videos like this, please give a like and a thumbs up. All right, and subscribe to the channel. If you want to see videos like this, write us in the comment, if any of the question paper that you want us to solve, we will give that a higher priority and solve it as soon as possible. See you in the next video. Bye bye.